Now, for Module 7.1, chromosome structure and karyotyping, the most important thing we have to take note of would be the levels of packaging as well as how to make a karyotype. So there are three levels that we discussed. The DNA would be the naked DNAs is without any kind of packaging, and then the first level is beads on a string, which is around 11 nanometers. So we'll explain that a little bit why it's beads, because of the presence of the histones. Then when the beads on a string will go around each other, you will have the 30 nanometer chromatin, which is also called the solenoid. And then you have here the 300 nanometer fiber, which is just like going round and round and round on a scaffold. And then you have higher packaging, and finally you have your chromosome. So that would be the most important thing you have to take note of here. So this is the naked DNA. This would be the beads on a string, as you can see here. It's like beads. When you put them together, you now have a hollow solenoid. And then you have here your 300 nanometer fiber and higher structures, and finally you have your chromosomes over here. So the nucleosome would look like this. So this is the histone core, and the H1 will be clipping together the DNA that went around it twice. So this is very important. And then of course the structure of the nucleosome contains H2A, H2A, H2B, H2B, two copies of each on the top portion of the octamer, and then the H4, the H3, H4 and H3 on the bottom portion of the octamer. So this would be the nucleosome, and the DNA will be held together by our H1. So that would be making sure that the DNA will be packaged, and of course it's very important because it allows a very long DNA to be packaged inside of a very, very, very microscopic tiny nucleus. So that's why this is very, very important. And the next thing is about karyotyping, and karyotyping is a way of arranging the chromosomes by making use of three important characteristics, which is shape, size, and the gene positions. You will be able to determine the chromosomes, or what type of chromosome, what number of chromosome would be by determining all of these three important characteristics. And we already know that the shapes of the chromosomes would be as follows. You have here since the centromere is at the end, this is the telocentric. This one is almost at the end, and you have a very small p arm. You have here the acrocentric. Then you have here not exactly, obviously not exactly on the middle, so you have the submetacentric, and this is more or less on the middle, so you will call it the metacentric. So these are the different shapes of your chromosomes. Now, very familiar with us. So if you have here chromosome 22, then you know that this is an acrocentric. You have X chromosome, this is submetacentric. You have chromosome 19, and it seems to be metacentric. And then you have here 15, 14, 13, which are all acrocentric. And chromosome number 1, which is also metacentric. Now, chromosome 4 will be submetacentric. So that's how you identify the shapes of the chromosomes. Alright, so it's just the same. So 17 will be acrocentric, 22 will be acrocentric, and then of course chromosome 1 would be metacentric, with chromosome 4 being submetacentric. Now, of course, you already know this, that the chromosome 1 to 22 are autosomes, which are not the sex chromosomes, and then the X and Y are the sex chromosome, which are important to determine the male and the female, XX being female and XY being male. So for the karyotyping procedure, we already discussed this as well. So this would be the taking a small amount of blood and culturing that, putting colchicine so that it will be arrested at the metaphase plate, which is the most condensed stage where the chromosomes can be obtained. And then the hypotonic saline is important for getting the cells to explode and releasing the chromosomes. Then this will be spread on a slide using trypsin to digest the proteins, leaving only the chromosomes here. So by taking a picture, you can now arrange that in a karyotype. Alright, so why are the chromosomes arrested at metaphase because it's the most condensed and easier to do a karyotype. Okay, so this is amniocentesis. By using a needle, you will get some fluid inside of the amnion to get amniotic fluid because it contains a lot of skin cells from your embryo, from your baby, so you can actually make a karyotype. On the other hand, the chorionic villi sampling allows a sample can be obtained from the chorion, which also contains blood from the developing embryo. So this is also very important for getting the karyotype of the baby. Very helpful. Alright, so what this order is shown by this karyotype, we discussed this already, so let's just go all over again. It is XXY, and this is called Klinefelter syndrome. Okay, you have here X0, and that is Turner syndrome, the absence of one of the sex chromosomes. You have here trisomy 21, which is Down syndrome, very familiar as well. And then you have here XYYY, which is only around 12 people have been discovered with.